Analyzing DNA is an extremely important technological process in the world. Industries such as the health industry, genetic research, forensics, and agriculture use the process of DNA analysis for performing tasks such as gene therapy, testing blood, taking DNA from crime scenes, as well as creating genetically modified foods, which are also known as GMOs. When scientists want to extract DNA, they only require a very small amount of DNA samples. Every cell in the body has DNA, therefore it can be collected via samples such as saliva, which is done in COVID tests, blood, and skin cells. After DNA is collected from the organism, the DNA can begin the extraction process. DNA extraction is a process that can be performed in a laboratory with test kits. The first step of DNA extraction is cell lysis. Cell lysis is the process of breaking open a nucleus, opening the membrane, which allows for the DNA inside to exit the nucleus. There are many ways and methods that can be done and used to perform cell lysis. It can be done physically by vortexing and grinding, which can be done by vortex grinding machines. Or it can be done chemically by using a salt and detergent solution such as sodium dodecyl sulfate. The salt in the solution protects the phosphodiester bonds in DNA, while the detergent breaks the phospholipid bilayer membrane of the nucleus. So these two methods, physical and chemical, can either be done separately or they can be done in combination with each other. The second step of DNA extraction is the purification of the DNA. So after cell lysis, the DNA solution may possibly contain unwanted debris such as proteins or lipids, or many more. So during the process of DNA purification, the debris is extracted. This can be done with enzymes, and an example of an enzyme used is proteinase K, which can degrade proteins. It can also be done by centrifuging, which is a method of filtration using a centrifugal or rotational force. By now, the DNA in the solution is a lot cleaner and more purified. The third step of DNA extraction is called precipitation. A salt and alcohol solution is used to precipitate the DNA. Since DNA is insoluble in alcohol and salt, it will precipitate in the presence of the solution. The precipitate formed will be a clearly visible white and stringy precipitate that can be extracted from the solution. And you can see in the image, that's what the DNA precipitate would look like. The fourth step of DNA is the cleaning of the DNA precipitate. So the DNA is cleaned and purified again by using wash buffers to remove any of the excess leftover nutrients, such as excess salt and proteins that were not removed in the second step. So what is left is a very purified DNA sample. The last step of DNA extraction is elution and conformation. The DNA sample is stored in a low ionic strength solution such as nuclease-free water. Nuclease is an enzyme that can cleave DNA, and it is an essential enzyme in the CRISPR-Cas9 tool. So now, the ready-to-be-used DNA solution can be tested and confirmed for the presence and purity of the DNA sample using a spectrophotometer. A spectrophotometer measures the light waves or photons that can pass through a solution. The DNA can then be grown and duplicated after it has been extracted. Processes such as the polymerase chain reaction and creating recombinant DNA can lead to the duplication of the targeted DNA that has been extracted to create mass amounts of targeted DNA. So to summarize, the process of DNA extraction follows the steps of cell lysis, purification, creating a DNA precipitate, cleaning the precipitate, and then putting the DNA in an elution and confirming for the purity of the DNA. After DNA is extracted and amplified by means such as the polymerase chain reaction, and then separated by the gel electrophoresis process, the DNA can be examined by looking at the base pairs, which is known as the genetic code. The genetic code composes up to 20 different amino acids in humans and can take form in a very large variety of polypeptide chains. By examining and understanding the genetic code, it can provide very important genetic information such as understanding mutations, gene functions, and quote-unquote normal sequences of DNA. Determining the sequence of bases is called DNA sequencing. This can be done through many different technologies. Some examples are the Sanger method, nanopore sequencing, or the whole genome shotgun sequencing. The Sanger method, which is also known as the chain termination method, was a process derived in 1977 for sequencing DNA. In this process, a DNA chain is terminated using a dideoxynucleotide, and the bases can be read through laser technology. The process of nanopore sequencing involves moving individual strands of DNA through nanopores, which are small pores or holes. And as the DNA moves through the nanopore, an electric current can pick up and specifically read bases, one base pair at a time. In the whole genome shotgun method, DNA strands are blown into small fragments, amplified using the polymerase chain reaction, and then by using computer technology, the sequences are reassembled. So when reading the genetic code, we can determine the genetic information that will eventually code for polypeptides through transcription and translation. This can be very helpful when determining mutations. For example, sickle cell anemia is caused by a point mutation in the DNA that codes for the protein hemoglobin. Hemoglobin is a protein that is important in the transfer of oxygen through the body. Normally, the sixth amino acid in a hemoglobin protein is supposed to be glutamic acid, but in people with sickle cell anemia, the sixth amino acid that is coded is valine. We all know this information because of DNA sequencing, and it is so important, and it can help scientists learn more about mutations and determine treatments and cures for genetic diseases. CRISPR is a gene editing mechanism that was derived from the natural CRISPR systems that bacteria cells have, which protects them from bacteriophage. In humans, the derived CRISPR-Cas9 system uses a piece of targeted DNA and a guide RNA. These components are found and synthesized in the nucleus of the cell. CRISPR must extract a healthy copy of target DNA from the nucleus to be used in the CRISPR-Cas9 system. Then after, the CRISPR-Cas9 system enters another nucleus, which will add the mutation to the DNA strand. That being said, the component of the cell that is important to the CRISPR-Cas9 system is the nucleus. The nucleus in a cell controls the activities that happen inside of a cell, such as metabolism, and it also carries hereditary information such as DNA and RNA. And here this is an image of the pathway that CRISPR-Cas9 takes into a cell. It enters into the nucleus and it uses its gene editing mechanism.